Hey everybody, Mr. Grove here. Today we're going to talk about meiosis. And so meiosis is what your body uses for the production of reproductive cells or sex cells, gametes. And so this would be sperm for the guys and then eggs for the ladies. And so this process of meiosis is where we go from a diploid cell down to a haploid cell. And so for the ladies, this takes place, basically prenatally, and all your eggs are there at the time of your birth. But for guys, you're continually going through meiosis and producing just billions and billions of sperm your entire life. And so this is kind of the process of how we do that. So you'll need to make sure you know the stages of meiosis. And there are kind of 10 is the bad news, but the good news is you've already kind of memorized these. And so just like our stages of mitosis were PMAT, that's also our stages of meiosis. We have prophase 1, metaphase 1, anaphase 1, telophase 1, then cytokinesis, and then prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis again. And so ultimately, in meiosis, we had one cell, and then we ended up with two cells that were genetically identical to each other, but then also to the original cell. But meiosis, we start with one cell, and then we end up with four cells, and we often call those four daughter cells. And those cells are haploid, so they are not identical to each other or to the original cell. And so these would be like the eggs or the sperm then, depending on if you were male or female. So let's go through the stages a little bit. So first we have prophase 1, okay, and so many of the same things that are happening in these stages are what was similar in mitosis as well. So in prophase, our nuclear envelope is going to start to dissolve away. Our mitotic spindles are going to start to form, and then our chromosomes are going to coil up and become visible. And then they're also going to pair up where the homologous chromosomes will find each other and then line up next to each other. It's kind of like in a little elementary school when you had to have the buddy system. You're going on a field trip and you had to find your field trip buddy. So that's kind of what's going on here. Metaphase 1. Those partners are going to form a double file line. So we're going to meet in the middle again. And so just like we did in mitosis, our mitotic spindles will be connected at the centromere to these chromosomes, and we'll maneuver them around to where we've, instead of a single file line, a double file line then for our pairs of chromosomes. Anaphase 1, we're going to pull apart. But in this case, we're not actually unstapling the chromatids, the simple sister chromatids. We're just going to pull the pairs apart from each other. So that one that came from mom is going to separate from the one that came from dad. The cell will start to elongate, and then we will also, the chromosomes will start their migration to the poles. And then in telophase 1, we're going to start to reform the nucleus. We'll start to unwind the chromosomes, and the mitotic spindles will start to pull apart, and cytokinesis will start to begin, and then we will actually have cytokinesis where we are now two cells. These two cells now would be considered haploid because they do not have a double set. They've got a chromosome from either mom or dad for each one, but not from both, and so we would now consider everything haploid. But we're going to go through it again. So prophase 2, mitotic spindles form, nucleus dissolves away, chromosomes coil up. Metaphase 2, we're going to meet in the middle, so those mitotic spindles will arrange and move those chromosomes into a single file line now. Anaphase 2, we're going to start to pull apart, and this time we are unstapling at the centromere, so that chromatid's going to this side, that chromatid's going to this side. And then in telophase 2, we're going to start the process of making two cells. The nucleus will start to come back together. Chromosomes will uncoil, and the mitotic spindles will completely wind up and start to disappear. And then our second cytokinesis will split these cells into two, and so now we end up with 1, 2, 3, 4 daughter cells that are all haploid. And none of them are identical to the other 3, and none of them are identical to our original cell that we started with. And so that is meiosis. And so if that was a guy, that would be formed 4 sperm cell. All 4 would be equally viable. And if that was a gal, that would be 4 eggs, and really just one of the 3 would form into a polar body or a viable egg, and the others would kind of dissolve away into non-viable eggs. So that's meiosis. Here's a couple more slides from just a different textbook that kind of take you through the different stages. So prophase 1, metaphase 1, anaphase 1, telophase 1, then we have cytokinesis, and then prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis again to end up with our 4 daughter cells. But meiosis is where we make you unique. You know, what makes you you? You know, why are you different from your siblings? You know, I've taught enough students in my career that I see similarities to other siblings within a family. And I've also taught long enough that I see, parent-kid similarities together within the same family. But why aren't we identical? Why aren't you a twin of your brother or sister? You have the genetic possibility, the same genetics from mom, the same genetics from dad. It's entirely possible, but we'll see that statistically it just does not become likely that you would have the exact same set of genetic DNA, even though you had the same mom and dad to give that to you. So the first one of what makes you you and truly unique is what we call independent assortment. So independent assortment is going to be back in prophase 1 when we had this random pairing up. So chromosome number 3 from mom found chromosome number 3 from dad, and they were side by side from each other. But what we did not tell them is who was to go on the left or who was to go on the right. So mom could have went on the left, or dad could have went on the left, and vice versa. And that happens for all 23 pairs. So for example, from a statistic possibility, if we have, let's say dad is blue and mom is red, so if we're just talking 3 pairs of chromosomes, we could have dad, 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 mom, mom, mom. We could have dad, mom, mom, dad, dad, mom. We could have dad, mom, dad, mom, dad. We could have dad, mom, mom, dad. We could have mom, 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 dad, dad, dad. We could have mom, dad, dad, mom, dad. And you can see all these different varieties. In fact, we would have 8 different possible combinations here. And so mathematically, that works out then that we would be 2 to the 3rd, because we have 3 pairs of chromosomes, and they could either be on the left or the right. So they have 2 options. And that turns out to be 8. And so we'd have 8 possible combinations of our chromosomes. Well, we don't have 3 pairs. We have 23 pairs. And so we would have, mathematically, 2 to the 23rd possibilities. And that 2 to the 23rd gives us a 1 in 8,388,608 chance of making the same identical sperm or making the same identical egg based on this random left-right lineup. Now here's the thing. Mom never even had 8 million eggs. She really only had like 1,000. And so the statistical likelihood of having 2 eggs that were the same are incredibly unlikely for her. Now, dad is definitely throughout his lifetime going to make 8 million sperm, but statistically still, the odds of having any 2 sperm exactly the same based on independent assortment would be 1 in 8,388,608. So highly, highly unlikely. And so that is independent assortment. Another thing that makes you you is this idea of crossing over. So back in meiosis, where we have prophase 1 and sometimes in metaphase 1, so we had that chromosome from mom pair up with that chromosome from dad. And it's all squished in there. So they match up, and so they're all kind of squished up. Well, this glue that glues all these genes together on the DNA is the same glue on dad's that glues all these genes together. And so it's really easy for it to become unglued and then re-glue. And so what happens is when anaphase starts to take place, these chromosomes will pull apart from each other. It's very possible then that we would get something kind of like this. So that little piece from mom accidentally got glued onto dad, and then that little piece from dad accidentally got glued onto mom. And so if this is the egg that ultimately gets chosen then, or if this is the sperm that ultimately gets chosen for fertilization, that's going to be a completely new genetic. That's mostly dad with a little bit of mom, and that's mostly mom with a little bit of dad. And so these new genetics or new genetic combinations would be very possible to be unique, that's never been seen before. And so that's what we call crossing over. So that happens in prophase 1 or in metaphase 1 when the chromosome pairs are kind of squished up and partnered up with each other, waiting to separate apart. Then the last thing that makes you truly unique is this idea of just random fertilization. So we said even if mom could make the same egg twice, which is possible, but it was that 1 in 8 million chance. The fact if you have 2 kids, you would have to pull those 2 eggs out of that large supply. And so, like we said, that started off with like 100,000 when you were being developed, and those eggs, just totally slowly dissolved away and became non-viable to the time you were of childbearing age, to maybe having a few hundred or a few thousand left. Dad, if he's going to make the 2 potential sperm that were identical, again, that's a 1 in 8 million shot, like we said, based on independent assortment, but the odds of that sperm being you and then your brother having the exact same 1 in 8 million sperm, between both of those being chosen to fertilize, that would be about 1 in 64 trillion for our odds. There are not 64 trillion people in the world. So the odds of being genetically identical to your siblings are not going to happen. So 1 in 64 trillion. Now, with that being said, you are truly unique unless you are an identical twin. And so that identical twin is kind of an interesting situation where we had a zygote form. So here's our zygote. And then when it went to split, that zygote should have just made 2 cells, right? But what happens is those 2 cells actually will split apart. And so rather than that first division sticking together, they are split apart. So here's twin 1, here's twin 2, and they are genetically identical. And so that's identical twins. Now, fraternal twins are not genetically identical. In fact, they're no more identical than just brothers and sisters. They just happen to be born at the same time. And so usually what happens is mom, rather than bring 1 egg down for fertilization that month, 2 eggs came down and 2 different sperm fertilized the 2 different eggs. And so that would be what we would have fraternal twins. Again, no more genetically identical than just any brother or sister combination. It just so happens to be that they were born at the same time. But in the case of identical twins, they truly are genetically identical because they came from 1 fertilized egg, and that first mitotic split, rather than sticking together, actually split apart. And so we have twin number 1 and we have twin number 2 that then continue to go through mitosis and develop into organism 1 and organism 2. And so that would be how you could be not unique to your sibling in the case of identical twins. So that is meiosis, and I hope that was helpful.